Morning, everyone. I'm very happy to introduce Edward here today. Where do I start? Edward and I actually share quite a long history. The first time I ever met Edward was in Panama in 2009, I think, when he was part of a course I taught where we had students from the University of Constance and Princeton students do a 3-week field course together. And he tracked bats with us. So even then, he was already looking at social foraging in bats. And then we kind of kept a loose connection. Edward kept on popping up in places, including our institute, while he did his PhD in Maryland with Jerry Wilkinson on the social foraging of the Mexican fishing bat. And I think he's going to talk about that a little bit very briefly today as well. And then he applied for actually a postdoc in the cluster. And I had advertised the PhD position based on the application that we submitted for the cluster. But then somehow it seemed like a match made in heaven, and it was decided that I was allowed to hire a postdoc instead of a PhD student. And that's how and why Edward joined our group. And I'm super happy about that. Edward hasn't been able to do fieldwork yet because of COVID and Icarus delays, but he has been super productive. And I think he's going to talk about that. And we look forward to fieldwork soon, right? And with that, I hand over to you, Edward.
Okay, thanks. Let me just share my screen. Okay, so yeah, thanks, Dina. Thanks for the introduction. I'm very excited to actually finally be in the office in the new VCC building and seeing so many people in person. And today I'll be talking about continental scale monitoring of Africa's most gregarious fruit bat. And just to go a little bit back into my interests, I'm interested in working on sort of the interplay of how resource availability and sociality influence animal movement. So depending on the landscape and how social a species can be, that can really change how species move around. As Dina mentioned, I'm just going to touch a little bit on my PhD work. So I was studying fish-eating bats in Mexico. So I was working on this tiny island, let me get the laser pointer working, called Isla Partida Norte in the Gulf of California in Mexico. And we were tracking these very cute little bats here and putting GPS and audio loggers on them and exploring their foraging behavior. And so it was really exciting to get to connect GPS tracking with acoustic monitoring because bats are basically screaming their behavior all the time. And so echolocating bats produce these very characteristic sequences of calls seen here as a buzz. And what we were able to do is identify where these feeding buzzes occurred and get a much more in-depth look at where bats were actually foraging during their nightly flights. And so we were able to then look at what type of habitat a bat actually cares about in the ocean, and try to understand how a bat evolved to try to catch fish, as well as looking at some of the social aspects of foraging, how the bats are actually using each other's feeding buzzes to then eavesdrop and find new foraging sites. And so this work was with Jerry Wilkinson at the University of Maryland in collaboration with Yossi Obel at Tel Aviv University. And I'd be happy to talk to any of you more in depth about this project at another time, but I'm gonna focus instead on my current work, which is looking at a much larger pattern of migration in fruit bats in Africa. And so I'll start here by talking a bit about the role of resources and how resources can move through the environment. We typically think about resources as kind of static, maybe a fruit tree, but both spatially and temporally, resources can really change. And so right now, a lot of the fruit trees are just beginning to have fruits. I went cherry picking last week, and it was quite lovely to get to see sort of this resource wave emerge. And so these resources can appear in certain areas very briefly, and they can range in a variety of scales. So if you've ever been to the neotropics, you might have seen an army ant swarm. These are waves of ants that move through the forest floor and basically push through, are pushing all of the insects. They're ripping apart all of the insects, but they create this front where all of the insects in front of the front are running away for their lives. And they create this temporary resource that birds can use, where essentially all of the insects typically are hiding, but when the army ant swarm comes through, they have to leave their hiding spot and run for their lives. And so it creates this unique resource that birds can rely on to catch insects very easily. And so this is sort of a wave that moves through the forest floor that many species of birds have evolved to strategically follow. In addition, we can see more mesoscale patterns, such as marine fronts, where you have different bodies of water, either with different temperatures or salinity, and where they mix, you often get upwellings, and that will bring nutrients up to the surface. And so many marine species actually strategically go to these fronts to forage. And then finally, we can think about snowmelt as a resource wave. A lot of species, don't have a good ability to forage in the snow, and so they're actually tracking snowmelt and moving along this gradient through the ecosystem for migration. And so we can actually predict how fast a species should be migrating based on the speed that snowmelt actually moves through the environment, and make some predictions about how species should actually be responding to this, be migrating accordingly. And we see that actually there are a lot of species that are tracking resources. And this typically occurs in environments that are seasonal. So if you always have food available, it doesn't really matter if a wave of resources comes through, because you can benefit from just staying in the same place. But if you're in regions where there's kind of a boom bust cycle with food, then you really need to track these resource waves. And here's just an illustration from a great paper in Tree, illustrating just the variety of species that have been studied so far that follow these resource waves. And they can be driven by different factors in the environment, such as temperature, elevation, precipitation, and wind. And so how those environmental variables interact with the landscape can create waves of resources that species can then try to follow. In addition, some of these patterns of surfing resource waves seem to have a bit of a migratory culture to them. And it's been suggested that emergent sensing might be able to aid groups in following such gradients. And what we see here is actually an accidental experiment where historical populations of sheep and moose were translocated from their original locations, and in some of the translocated individuals, they actually lost the ability to correctly surf a wave. And so it's possible that, this green wave surfing or resource tracking through the environment can be lost if you have sort of loss of this information in the population. And so it suggests that there's a strong social aspect with some species to how they're able to follow these resource waves. So I'm interested in looking at patterns of animals following resources that we might not necessarily be able to track using standard measurement techniques from satellites. A lot of the species that I talked about were responding to changes in greenness of the landscape, and they're directly eating that greenness in the landscape. They're eating leaves of plants, or species that might be responding to temperature, and you can see that snowmelt is changing. But instead, if we're looking at things like fruits and flowers, that's less obvious how you can actually study that in remote sensing data. Additionally, most of the studies focus on more temperate regions where seasonality is a lot more dramatic. And I wanted to look and see if this pattern really holds up in the tropics. And in particular, I'm focusing on Africa. And on the left, you can just see the greenness cycling up and down across the equator each year. I'll talk a little bit more about that in a second. But first, I would like to introduce our study species. This is the straw-colored fruit bat, Eidolon helvum. And they are a generalist frugivore. They eat a wide variety of plants. And so they'll consume, I think, over 200 species of fruits and flowers, and even some bark and leaves. And they're found throughout much of sub-Saharan Africa. And these bats also form large colonies in the thousands to millions. And these colonies can be in remote regions or in the middle of cities. And we actually don't know too much about the natural history or biology of this species, mainly because it's hard to actually observe them too closely. When you get close to a flock or a roost of these bats, they tend to fly away. And so it's very difficult to actually understand who is roosting together and how they're foraging in these environments. But Dina's lab has already done some very impressive work looking at these bats and how important they might be for the ecosystem. And so one interesting thing about bats is they're often pooping while flying. And so if they're consuming small seeded fruits, then they're actually digesting and passing those fruits while flying. And so there's this large potential for seed dispersal, where they're actually raining seeds across the landscape as they're flying. And this could potentially be a very important factor in reforestation in areas across Africa. In addition, we've been monitoring some of the colonies, and I'll talk about that a little bit more in a second. But one trend that we've been seeing is that a lot of the colonies, or some of the colonies, have been showing precipitous declines. And so it adds sort of a sense of urgency to understanding their migration in these aggregations. And one of the threats could be from sort of the bushmeat trade. There was a study that just focused on southern Ghana, but estimated that over 100,000 bats are sold each year, which is not believed to be a sustainable number of individuals. And so that could be one of the main causes of this decline in Accra. And so because of that, we've been working on this Eidolon monitoring network, where we have volunteers across Africa in as many places as we can find. And if you know of any people that might be interested, we'd love to find more. But these are volunteers that will do monthly counts. And these are systematic counts, all using the same method to estimate how many bats are in a population. And this was started by Jakob Farr and currently run by Natalie Weber. And you can follow us if you're interested on Twitter at Elon Network, and also check out our new website at eidolonmonitoring.com. And so the idea is to make all of this data open access and hopefully hand off the project to environmental groups in Africa to be continued, so that they can sort of take ownership of monitoring their species. And so I've been analyzing some of the data that's already been collected from this project, and we're trying to understand the dynamics of how these bats might be moving across Africa. So let me just give you an example of what the data might look like. So we can plot the number of bats, the relative number of bats in a colony across the year. And what I did was simply look for the peak. When did we have the most bats appearing in a colony? And then I wanted to see if there was remote sensing data from satellites that might give us information about why the bats might be at their peak at this time. So here is EVI, which is known as the Enhanced Vegetation Index. It's a common metric for just measuring the greenness of the landscape. And another metric that's often used in addition to EVI is looking at the first derivative of EVI. So this is called the instantaneous rate of green up, or IRG, but it's essentially telling you the rate of change of EVI over time. And so from both of these, we can pull out when do we see the highest or the peak greenness of the landscape in EVI, and we can see when do we see the peak IRG, or peak rate of change in a landscape. In addition, we can also get measurements of precipitation for an area. And we can say, okay, when is the rainy season at its strongest in a given region? And essentially what I was interested in looking at in the data is, are there patterns in any of these phenological events? So peak greenness, peak rate of change in greenness, or peak precipitation, and do they commonly occur close to the peak in colony size? So this is just, again, this animation of this greenness moving up and down across Africa. And one of the reasons that we thought that bats might be responding to this phenology is because when you look at what's called the start of the season across Africa, you see these very distinct bands. So theoretically, a species could be moving along these bands, and if their food source is occurring with one of these phenological events, that could be driving their migration up and down the continent. You might also notice that there's missing data in the middle, in the Congo Basin. And that is partly because of cloud cover. It's hard to get good estimates of greenness changes, but also because there's less seasonality, there's less change in the environment over time. Although you do typically see that fruiting events do still have one annual peak, even in the Congo Basin, that's sometimes a little bit harder to detect in the data. And you can see this again in this metric of seasonality. Here, perhaps we can start with the figure on the right, is what's called entropy. And this is a metric for defining how seasonal the environment is. And all it is calculating how much of the year that greenness is spent in just a single month. So if you have extremely high entropy, that means that you just have this one peak in greenness during the year. And then the rest of the year, it's very brown. Whereas if you have very low entropy, that means that you have relatively stable greenness across the year. And so we can then map that onto the continent of Africa and see that sort of these subtropical regions tend to have much higher entropy or seasonality, where you have these explosions of abundance of resources for very short periods of the year. Whereas in the Saharan desert, there's not really much happening, but it's consistently dry and desert. And in the Congo Basin, it's consistently green. And so you also don't have high entropy or seasonality. So with this data, we wanted to hypothesize that colonies likely match the peak colony size with some seasonal explosion of resources. And so this could potentially be driven through collective sensing. And so what we would predict to find in the data is that at least one of these phenological variables that we're measuring will consistently be matching what the colonies are doing. Additionally, we would expect that there's a closer match between colonies that are in more seasonal landscapes than, say, in the rainforest, where there isn't as much seasonality. And additionally, we would expect that colonies that are larger would have a much better ability to collectively sense and track resources through the environment. So here I can give you an example of what some of the data, the true actual real data looks like. And this is from Accra, the site that I talked about earlier that was having this large decline in the number of bats. And Kasanka, this is a very famous bat roost in Zambia that is actually known as the largest bat colony in the world. And so we can then match up the peaks in colony size with the peaks in these different phenological variables. And just to give you information, that the colony counts are typically monthly, as well as precipitation is monthly, and EVI and IRG are bi-monthly. So we can then look at just a simple circular correlation in the data to say how well is the month that the colony is at its peak matched with the month that these different phenological variables are at their peak. And interestingly, EVI and precipitation have a very low correlation with when the colony is at its peak. And we actually see the strongest match with this instantaneous rate of green up. And here is just plotted the residuals of that plot as violin plots. And what I'll be doing next is trying to explain some of the variation. So why are some colonies closer matched for some months and other colonies further away? And so to do that, we took the absolute value of those residuals to then see how that would vary with different environmental variables. And so we find that closer matching is always at lower for these plots. So this is essentially the difference in months between when the colony is at its peak and when this phenological event happens. And that colonies have a closer match or a better match with IRG when areas have generally really high greenness or max EVI. Additionally, you also see this pattern that in more seasonal areas, you also have a closer match. And then finally, we also find that larger colonies have a close match. And so this is sort of consistent with our predictions and suggests that these colonies are matching this green wave in these seasonal environments. And here is just a plot showing the IRG for the whole continent of Africa divided by each month, and then red dots for when each colony is at its peak. And we see that a lot of the colonies are actually hitting this yellow line as it moves across the continent. But we don't actually know what the individuals are doing. And so our next step is really to look at long-term tracking using Icarus tags. And our goal is to really test if bats are indeed surfing the green wave. And what we're hoping to do is tag at 5 locations across the continent of Africa and as divided as possible across a longitudinal gradient so that we can see at multiple points along this green wave, are the bats staying with it consistently. In addition, we also hope to find new roosts. We predominantly know about roosts in major cities, but the bats are moving all across the continent, and there could be locations that we have yet to find. And this will also give us a better understanding of the connectivity between roosts. And are bats simply going from one large roost to another, or is there something else happening in between? And then finally, we also hope, once we have a better understanding of where the bats are spending their years, we hope to go to certain locations and try to strategically capture mothers and pups, and maybe look at some of these factors of how the migration is developed over time in individuals, and could that be related to the migration of their mothers. So far, we actually have some recent tagging in Rwanda. We were able to collaborate with the Rwandan Wildlife Conservation Association, and Innocent here was kind enough to send us photos of him putting Icarus tags on bats. And we already have data that was sent from the tags to the International Space Station to my computer, which is fantastic. And we can see that the bats were tagged here in the southwest of this lake here in Rwanda, and some individuals have actually traveled around the lake. So this is still very early, but we hope that the tags will continue to perform well, despite the bats being in these very large roosts, sometimes under canopies. These are solar powered tags, and so ideally they will record for the lifetime of the animals, but we're still in the piloting phase of this. But with the tags so far, we are hoping to tag quite a number of individuals over this year and next. And it's actually incredible that Icarus tags are so light. They're about 5 to 6 grams. And these bats are 250 to 300 grams. So we essentially can tag any individual that we catch. And what we're hoping to do with that is actually get a good cross section of the population. And we'll be able to sort of identify individuals that we tag by taking different samples from them. So the first thing we're really interested in is actually taking wing punches. So getting a small tissue sample, you can then look at the age of an individual. So there's a new method called DNA methylation analysis where you can actually see how changes in the methylation of a species lead to changes in their age. And using a calibrated clock, which there just so happens to be a calibrated clock for this species, you can accurately estimate the age of an individual just from DNA methylation within about a year. So then we'll have some idea of maybe the experience of migration for individuals, despite not tracking their entire life history. In addition, we hope to collect some poop just to get information on the diet of these individuals. We'll be using DNA barcoding, as well as looking at viruses. We hope to understand how viruses might change in a population of bats, especially how migration might influence the viral load that species have over time. And so being able to perhaps catch and sample and see how bats viruses change depending on where they are on the migration route would give us a lot of insight into how bats could potentially be a vector for spreading viruses. And then we're also hoping to look at nutritional status of bats as a potential driver of migration in certain instances. We want to understand if bats are leaving areas because they're maybe nutritionally deficient in some resource, and that might be driving them to leave, or alternatively, they could be predicting that there's some new bountiful resource elsewhere that they want to leave early where they are to go meet. So there's a lot of high hopes for what we can find once we start tagging more bats, and I can't wait to tell you guys about it. And I just want to finish by talking a bit about another project that I've been working on with Ben Koger to actually automate colony counts. So all of our methods previously have been using observers to estimate how many bats are flying by. But we hope to sort of develop a more quantitative method for this. And so Ben is using deep learning and videos. So these are just simple GoPros that are pointed at the sky around bat colonies. And we can then use that to track and count all of the bats that fly through the field of view of the camera. And so for this video, the roost is at the bottom of the screen, and they're going to forage towards the top. So we expect all of the bats to be flying up across the screen. And as they cross the center of the video, they're counted through Ben software. And so this is just a schematic of how the cameras were placed. And this study was done at Kasanka National Park, which is, as I said before, the largest bat colony in the world. And we placed about 10 cameras around the park. And as the bats flew over, we could estimate the number of bats flying past the field of view of each camera. So these gray dots are the locations of the cameras, and the lines represent sort of the field of view, the maximum field of view the camera should get. And then we weighted sort of the area or the angle of departure that we would expect to cover by each camera. And so putting all of the counts together, we can then create estimates of how many bats are flying out of the roost each day. And so we see then changes between days, as well as we're able to get sort of an average over multiple days of how many bats might be in a given roost. And what we're hoping to do is look at these count numbers across years and across multiple timings during the bat occurrence to really get a much more robust estimate of how many bats we expect to be living in this colony. And one very interesting facet of this research is we can get behavioral measurements basically for free using this method. In addition to density of bats in the field of view of the camera, we can also look at their polarization. Are the bats actually moving in a coordinated movement together, or a bit more haphazardly across the screen? In addition, we can pull out wing beats from the individuals. We're constantly measuring the size of their wingspan to get an estimate of their height. And so both wing beat and height information can be very useful for understanding how the bats are leaving their roost to go out foraging. And so we're hoping to use this data to better quantitatively measure colony size. We really want to understand how colonies sort of rise, peak, and fall over the season that they're around. And we could use this method to more accurately detect these changes. And that's something that we currently can't really get from monthly counts. You don't really see dynamic changes in the number of bats. Excuse me. And perhaps if the bats suddenly arrive or suddenly depart, that could signal some sort of collective sensing of the environment. And that's something we would first need to detect before we could really explore better. In addition, we also hope to have more information on how colony sizes are changing between years. Are colonies consistently decreasing as some colony counts suggest? And one of the factors that could be interesting with this is if the bats are actually doing collective sensing, there might be Allee effects or density dependent behaviors that could be influenced by a rapid decrease in population size. So if the bats are relying on each other to know when to migrate and know where to find food, then a rapidly decreasing population could be catastrophic. And so we really want to understand in a more quantitative way how colony sizes are changing to really understand if the population is at risk. And this is all in the context actually of two recent impending threats to Kasanka National Park. The first is an industrial agricultural site that's being planned within a protected area, but just outside the border of the park. And this would cut down many fruit trees and instead plant a crop, as well as divert a lot of water from a nearby river. So it has a lot of potential effects on the park itself and the bat community. And there's currently a campaign at kasanka.com to protest the construction of this. As well, there's also a wind farm that's being proposed to be developed within about 60 kilometers of this bat forest. And while most work on wind turbine interactions with bats has been on much smaller bats, fruit bats are also at risk of colliding with wind turbines. And from what we see with some GPS tracking data from this colony is that the bats reliably forage about the same distance that the wind turbines will be placed. And what I have are different rings from different studies of estimates of the average foraging distance. And essentially all of these average foraging distances encompass where this wind turbine will be placed. So it's definitely something that we need to keep an eye on and could potentially affect the bats dramatically. So with that, I would like to thank you for your time and thank all of my many collaborators through Wandin Wildlife Conservation Association, Kasanka Trust, Dina, Natalie, and Ben, and the University of Constance, Max Planck, and on the right are many volunteers that have helped us with these colony counts over the years. And with that, I'd be happy to take any questions.
Hi, can I have a question? It's an exciting project. Can you hear me?
Yeah. Hi.
And then, so I wonder that how you distinguish between collective sensing and some hardwired or innate mechanism. You know that if bird increased suddenly in a certain region, that could indicate either collective sensing or anything else, maybe hardwired or something. So what's your strategy of distinguishing the ungulate migration culture that they are recently migrated and then that they are efficiently improved over a short time, right? Mm-hmm. And then that's something like that. What do you have to support?
Yeah. So that's a very good question, and it's very difficult to do experimental work with the species to really manipulate their environment and put them in a scenario where you could directly test that. So right now we're mostly focusing on correlative features such as how bats in different size colonies would respond to changes in the environment. And so if you're in a very large colony, we would expect that you would be better at detecting these resource waves than in a small colony. And in addition, we're hoping to see if this resource tracking is also a byproduct of age, and that individuals who are older might be better equipped at following these. But yeah, if you have specific ideas for how the bats, how we might be able to experimentally do this in quite a challenging system, I'd be happy to talk with you more about that.
So how many bats are you planning to tag, and what percentage of bats would you need to get a good estimate of what the whole population is doing? And a related question, if you take the Icarus tags, I think they give you 1 or 2 positions a day, and you showed that their range is very large. So are you selecting the time of the day when you want to get the position, or how do you cope with this large range that they have every day and your few positions per day on the tag?
Yes, so for this project, we'll be looking just at 1 GPS fix a day, and that will be around noon, so that we get their roosting location. And that way we can sort of consistently be measuring where the bats are roosting over time. I think our plans so far are to tag about 150 bats this year. And we're hoping to divide that among all of the 5 sites as best we can. So having roughly 30 tagged bats per colony will give us some indication of how they're changing. But yeah, it's a good question. What's the sort of ideal number to tag? We don't actually currently know the full migration route for any individuals. And so I think once we start to get some data back, we'll get a better understanding of the variation that happens within each colony and more precisely be able to see, are they generally doing the same thing? Or is there a large variation among individuals? So it's a little too early to make firm statements about this is exactly how many we need. Dina, you have a question?
Yeah, I was wondering, but I know you've thought about this, but whether you could talk a little bit about how IRG relates to actual food availability and how you, because it's kind of the increase of food and maybe precedes food peak abundance.
Yeah, so it is a little strange to think about fruiting occurring before you have sort of peak greenness in the environment. And I think that potentially what's happening in a lot of places in Africa is you have greening happening actually before the peak in rainfall. And so this is a phenomenon that's been found across much of Africa, is that trees are actually trying to get all of their growing and flowering and maybe fruiting done before you have these seasonal rains. And so potentially fruit species might be occurring on trees that are already large and adult sized, and it's simply the trees that have been saving resources, and then they sort of let out some resources before the seasonal rains occur. But it's definitely something that we hope to look more directly at. We want to sample the diets of the bats in different regions and then more strategically look at what species are occurring there. Right now, this is all very correlative analyses from satellite data, but once we can actually go to places and start looking at what the bats are doing, we'll have a much better idea of what's going on. So Kriesler asked, he was wondering, what is the extent of hunting as a threat to migrating bats in the region? And is it possible that tagging and tracking bats would give us information on their vulnerability to hunting? That's a really good question. I haven't directly thought about that. I think it would depend on sort of identifying how a bat dies or stops recording. We would need to communicate with people in the field to try to recover tags that stop functioning, and then try to identify the source of death. But it is something that we hope to do is talk with people in each region that we're tagging and better understand sort of the impact that people are having on bats in the regions. And so just from people through the monitoring network, we know that these bats do cause a lot of problems. Essentially, they're extremely loud in their roosts, and people don't necessarily like that. And so people will clap or approach the roost just to get the bats to try to fly away. But in general, what we know is that the bats tend to roost in areas where they're less likely to be harassed consistently. So even when they're in cities, they're often in hospital grounds or embassy sites or golf courses where I guess the general public is not as free to roam. And so that's where the bats tend to find a bit of safety. But we definitely know that people, if bats show up in their garden, maybe the people will cut down their trees because they don't necessarily want the bats around for months. Lucy has a question.
Hi, really interesting work. I was just wondering, maybe I missed this, but when you were talking about threats, it reminded me that recently in Australia, there's been a few mass die-offs of bat roosts, fruit bat roosts to do with temperature. So it seems to be, days above 45 degrees or something can you get mass death. And this is obviously linked to climate change. Is that another potential threat that you could actually look at with these guys?
Yeah, I definitely think it's something that we should keep in mind and have sort of an analysis to see. Yeah, I haven't seen any news reports of that happening in Africa, but that doesn't mean it's not happening. And the bats definitely will show up in extremely warm places in the Sahel, or I've even seen reports of, potentially vagrant bats showing up in Namibia. So they can encounter extreme heat, and understanding how that influences them as well would be definitely interesting.
It sounds like they're perhaps more able to move, because I find this quite a strange phenomenon in somewhere like urban Sydney, that the bat roost doesn't appear to be able to move even though they fly a long way to forage, the roost doesn't appear to move in response to temperature threats.
Yeah, it seems that these bats are extremely mobile and can simply relocate if there's too much of a problem.
I have a question. Really nice talk, Edward. Looks
Thanks.
super cool. I was curious whether you are, well, I was curious about these periods where there's a trough in the colony size. And I'm wondering if you're, it sounds like, you know, one of your expectations is that maybe the trough is at this time point is occurring because all the bats are at a peak at a different colony site. But I was wondering if, I guess maybe an alternative way they could be surfing the green wave sort of would be to be very dispersed during these low periods and then kind of congregating during the peak in food availability. So I was wondering if you think that might be happening, or if you have a strong sense about which of those two is happening and if you'll be able to detect it.
Yeah, I think we would definitely need people on the ground to work with, but that is one of the proposed hypotheses, is that the bats kind of disperse into the rainforest, into the Congo Basin during periods of time when they're not more concentrated in seasonal landscapes. But we also have data from an individual tracked from Kasanka that simply crossed the Congo and showed up on the other side in South Sudan. And so it's possible that bats are just migrating really far and going to another seasonal landscape to exploit that, and really following the green wave across the rainforest. And so, yeah, we really don't know what to expect when we start tagging individuals, if they will sort of disperse more. It's possible that females giving birth will respond differently than males, and they might seek out more secluded regions for that. But it's something that there isn't really good data on for what the bats are doing in the rainforest, mainly because we don't find these very large density colonies over there.
Thanks.